All right, let's talk about the most amazing recipe book in the universe, the one that's inside every single one of your cells. Today, we're going to figure out how a cell actually reads its own DNA to build everything it needs. So let's jump right in.
Okay, so we've all heard it a million times, right? DNA is the blueprint of life, the master plan. But have you ever stopped to wonder how that actually works? I mean, if it's the priceless master copy, how does the cell read the instructions without risking any damage to it?
Well, here's a great way to picture it. Imagine your DNA is like a massive, secure library filled with priceless, one-of-a-kind cookbooks. These books are locked away for safekeeping in the cell's nucleus. They contain every recipe your body will ever need, but you absolutely cannot just check them out and take them home. They're way too valuable. So what do?
The cell's solution is just brilliant. It makes a copy. This process is called transcription. Instead of taking the original, fragile cookbook, the cell just copies the single recipe it needs, one gene, onto a small, disposable note card. That note card is a molecule called RNA, and it can safely leave the nucleus and carry the recipe out to the kitchen.
Now, to make a good copy, you need to know exactly where the recipe starts and where it ends, right? You can't just start copying in the middle of a sentence. So, let's take a look at the signposts written into the DNA itself that tell the cell's machinery where to go.
That specific chunk of DNA that holds one complete recipe is called a transcription unit. You can think of it as a single complete chapter in that giant cookbook. It's got all the information for one specific task. And each one of these units has three key parts. First, there's the promoter. That's the big, flashy Start Here sign. Then you have the structural gene, which is the actual recipe itself, the part that gets copied. And finally, you've got the terminator, the stop sign. It's that simple. Start, copy, stop. Incredibly elegant.
Okay, this is where it gets really clever. Remember, DNA is a double helix with two strands. The copying machine only reads one of them. We call that the template strand. The other strand, the one that's ignored, is called the coding strand. But here's the cool twist. Even though it isn't read, the coding strand sequence is almost a perfect match for the final RNA copy. It's a bit weird, but that's how it works.
So, we know the layout, we know the markers, but who actually does the copying? Well, let's meet the star of the show, the tiny molecular machine that acts as the cell's master scribe.
The whole thing plays out in three clear steps, just like our start, copy, stop signals. First up, initiation. The main enzyme, called RNA polymerase, finds and latches onto that start sign, the promoter. Then comes elongation, where the polymerase just zooms along the DNA, building the new RNA message as it goes. And finally, termination. Once it hits that stop sign, it lets go and releases the brand new RNA copy.
But RNA polymerase doesn't work alone. It's an absolute pro at the copying part, but it needs a little help finding the exact starting line and finish line. So, in bacteria, it has some helper proteins. An initiation factor, called sigma, acts like a guide to help it find the promoter. And then a termination factor, called rho, helps it spot the terminator and know when to stop.
Now, this super streamlined process we've been describing, that's how it works in simple organisms like bacteria. But in more complex life, like us humans, we're eukaryotes, the process has some extra, really fascinating layers. It's kind of like the difference between a food truck and a Michelin-starred restaurant kitchen.
As the source material itself says, in eukaryotes there are two additional complexities. And this is where it gets really cool, because these extra steps give our cells a ton more control over which recipes get made and when.
This table really lays out the big differences. In bacteria, transcription happens right out in the main cell fluid, the cytosol, and the recipe gets used right away. In us, it happens inside that protected library, the nucleus. We also have at least 3 different kinds of RNA polymerase for different jobs. And, most importantly, our RNA recipes have to be edited and processed before they're ready.
And that editing step is the final, crucial stage for us eukaryotes. You should think of that first RNA copy as a rough draft. All the information is there, but it needs to be cleaned up, polished, and protected before it can be sent out of the nucleus.
One of the wildest parts of this editing is called splicing. See, it turns out our genes have the useful bits, called exons, and then a bunch of junk in between, called introns. Splicing is the process of precisely cutting out all of that intron junk and stitching the important exon pieces together into one clean final message.
So the whole editing workflow has 3 parts. First, capping, where a protective cap gets put on the front of the message. Then, tailing, where a long tail is added to the back to keep it stable. And finally, splicing, where the junk introns are removed. All these steps make sure the recipe is perfect and can survive its journey.
And what do you get after all that work? A fully processed, mature messenger RNA, or mRNA. This is the finished product, the perfect recipe card ready to be shipped out of the nucleus and into the cytoplasm, where the protein-making machinery is waiting to read it.
And that leaves us with a really fascinating final thought. This whole complex editing thing, this splicing, it seems almost too complicated. Why have the introns at all if you're just going to cut them out? Well, some scientists think this might be a ghost from our planet's deep past, a relic of an ancient RNA world, where RNA itself was the original master molecule of life, long before DNA ever came along. A little puzzle about the origin of life hidden in our own cells.